Hello everyone, myself Anush Dole. Today we will be looking at an overview of a biomolecule named lipids. Through this video, we can able to get information about lipids, their structures, how they bond to each other, their classification, even their roles in metabolism, along with the techniques that can be used to study the lipids. So let's start this video now. So let's start. What are the biomolecules? Biomolecules are then organic compounds that can be known for life forms. There are four different types of biomolecules named carbohydrates, proteins, nucleic acids, and lipids. They are like the four walls of our life. They play an interesting roles, yet they are connected to each other, maintaining the cellular structure and functions. Like they are the essential ones for our life. Study the different types of biomolecules in the next slide. So yeah, here are the different types of biomolecules, namely nucleic acid, lipids, carbohydrates, and the proteins. The first one is nucleic acid, which contains DNA and RNA. So this is the genetic basis of our life, which contains genetic material, and that is responsible for all the genetic transfer as well. We can identify a person by their genetic basis, which mainly contains this. The second one is lipids. Lipids are like the fat storage in our body that are even used for the cellular structure that is made up of them. The another one is carbohydrates. The carbohydrates are mainly sugars like monosaccharides, polysaccharides, and the oligosaccharides. So these are the immediate energy source that can be accessed through the body. The another one is proteins. Proteins are formed by peptide linkage from amino acids, and these are the essential for any enzymatic activity that can be done. So we will move further. So we'll now go with the significance of biomolecules in our life. Like we can say energy source and the storage. Lipids that are the energy source and acts as the storage as well. Another one is building blocks of life. Building blocks are like the pillars of our life, which if one of them collapses, our life won't be as regular as it is. Like if we want to function our body well, all the four biomolecules have to work interconnectedly good. Genetic information and storage that can be done by the nucleic acid, namely DNA or RNA. Structural support and the cell functions. This is the main primary work of the lipids that forms a structural support, likely a phospholipid bilayer 2. Regulation in metabolic and cellular processes that works for the proteins, even the cell signaling and communications too, along with the defense mechanisms like proteins like antibodies works differently for the defense mechanism. So each and every task that has been performed in the body has been performed by them, which is very essential for us to achieve a good health. So let's move to the further topic. So let's now focus on the lipids. So lipids are hydrophobic or amphipathic molecules, meaning they repel water or have water repelling properties along with the water attracting regions too. The lipids are like the fats, oil, waxes, phospholipids, and steroids. And the lipid structure are not the polymers like the other biomolecules like proteins and nucleic acid. The lipids are the small molecules grouped together due to their property of water insolubility. The lipids are insoluble in water, but they perform a crucial role in storing energy, forming biological membranes, and functional as signaling in the cell molecular processes. So, yeah, their roles are both structural and functional. Let's start to explore that in detail. So let's study the structure of lipid, like the basic structure of lipids. that is available throughout the life. So the basically the lipids are made up of two components majorly, that is glycerol and the fatty acids. Glycerol is like a three carbon atoms attached to three hydroxyl groups. This togetherly forms a glycerol group. Along with that, the fatty acids, which are made up of the acid group, which is attached to a hydrocarbon chain that is represented by R. So this togetherly form a structure called as lipids. Yeah. So lipids are classified into different types based on their structures like them, which is we will be studying in the next slides. So let's move on to that. So yeah, the as I have mentioned earlier, the lipids are classified into different types on basis on their basic structures. So yeah, the basic three types of lipids are simple lipids, compound lipids, and the derived lipids. The first one is simple lipids, in which the fatty acid is attached to alcohols. And the examples are like THC and waxes. THC is mainly triacyl glyceride, which is also called as triglycerides. This is the main component stored in the adipose tissue of the body that works like the energy source. Waxes could be like the wax extracted from the honeybees. Yeah. So second type is compound lipids, in which the basic structure of these lipids are like the fatty acids plus alcohol and the additional group like there are three different types like phospholipids, glycolipids, and lipoproteins. The phospholipids is like the additional group is of phosphate group. Glycolipids may you can see you can see the lipids are attached to carbohydrates that works for the energy source, and the lipoproteins, in which the proteins is attached to the lipids. The third type is derived lipids. It is basically the derived lipids are formed due to the hydrolysis of some simple lipids or compound lipids. That is mainly fatty acids, steroids, and eicosanoids. The fatty acids are further classified into two types based on the double bonds form in them. Like the saturated ones don't have any kind of double bond in their carbon atom chain, whereas the unsaturated fatty acids may have double bond between the carbon atoms. The unsaturated fatty acids are classified into two different types: monounsaturated fatty acids and polyunsaturated fatty acid. Like the monounsaturated depicts there is only one double bond. For example, the structure of folic acid, and polyunsaturated means there are more than one double bonds present in the fatty acid structure. Example, linoleic acid. Another type of derived lipids is steroids. For example, cholesterol. The cholesterol is very useful in the cell signaling as well. And the other third type is eicosanoids. For example, prostaglandins, leukotrienes, and lipoxin. So yeah, let's go ahead. So the compound lipids are further classified into three types named phospholipids, glycolipids, and lipoproteins. The first one is phospholipids, in which the basic structure is attached to a phosphate bond. The glycolipids are like the fatty acids are attached to carbohydrates to it, and the lipoproteins are like lipid plus protein wala bond. Then the phospholipids are further classified into two types named glycerophospholipids and sphingophospholipids. The glycerophospholipids contain glycerol as an alcohol, and the sphingophospholipids contain sphingosine as an alcohol. The glycolipids are also further classified into cerebroside and gangliosides. So yeah, that's it from the classification. Let's move from the for the next slide. So lipids also acts as the precursor for some hormones like the cholesterol. Cholesterol is a type of steroid which acts like a precursor for the steroid hormones like cortisol and aldosterone. Along with that, it also acts as a precursor for the sex hormones like estrogen and progesterone. So let's start with the role of lipids in the metabolism. The first one is beta oxidation of fatty acids. Beta oxidation is the process in which the fatty acids are broken down to get the energy source. So basically, it starts with within the cells. The structures stored in the form of triglycerides in the cytoplasm must be activated uh, to start the beta oxidation process. So for that, we need a coenzyme A that activates the fatty acids, and then the fatty acids are need to be goes inside the cell to start the beta oxidation process. A carrier has been introduced to transfer the lipids. The further step is like the dehydrogenation. Like the hydrogen atom from the structure has been removed to get an energy sourcing at a molecule like FADH. The further the water is added to that molecules. Along with that, another H hydrogen removal must be take place, which results into the formation of NADH. NADH and FADH are like the energy molecules that can be used uh, to produce acyl CoA. So acetyl CoA basically enters into the Krebs cycle and produces energy. Yeah. Yeah, so now let's study the role of lipids in the cellular processes. Everyone knows about the fluid mosaic model of the cell membrane, which depicts that the cell membrane is made up of phospholipid bilayer. The phospholipid bilayer says that it is a bilayer made up of phospholipids. So phospholipids are mainly made up of hydrophilic head and hydrophobic tails, in which the hydrophilic head is composed of phosphate groups along with the glycerol groups, and the hydrophobic tails are made up of unsaturated fatty acids and saturated fatty acids, which gives the amphipathic nature of the lipids. And the this phospholipid bilayer plays an important role in our body. Basically, the phospholipids creates a stable and separates the interior cell from the external environment. Another one is membrane fluidity. So lipid composition directly influences the membrane fluidity, like the unsaturated fatty acids in the tails, preventing the light packing and increasing the fluidity. Another one is the selective permeability. So selective permeability is like an barrier to some uncharged or charged molecules, basic on them, and membrane curvature. The phos Yeah, this phospholipid bilayer is essential for the vesicle formation that can be formed during endocytosis and exocytosis. And the last one is the platform for protein functions. Like lipid provides a functions like an environment to membrane proteins, anchoring and orienting the transport, signaling, and other enzymatic activities. So yeah. So let's understand the role of lipids in the energy storage, the main purpose of the lipids. The fatty acids are stored in the adipose tissue of our body as a form of triglycerides, which is the major source of an energy uh, in animals too. Like it is high energy density. Lipids provide an around 9 kilocalorie per grams energy, more than double that of carbohydrates and proteins. Like in comparison to that, lipids are more storage in energy density. It is efficient storage. Like uh, we can say it is stored in the triglyceride forms. It requires a less space and water so that it can store more and more energy. And it is a long-term reserve. Like it is an ideal for long-term energy storage during fasting or starvations. So yeah, that's the main storage energy role. So yeah, let's understand how the different types of techniques can be used to study the lipids, that is an essential molecules. So yeah, the first one is adsorption chromatography. Adsorption chromatography works to separate the lipids on the basis of the polarity of the lipid molecules. So basically what happens is uh, the some say stationary phase is added to a column, like the stationary field would be a silica gel, and then a sample with lipids is loaded. Then after that the mobile phase, like the solvent, is poured on the top of the samples so that it can be passed through it, which is called as the mobile phase. Then the lipids are separated based on the polarity of it, like the most polar lipid molecules stick to the silica gel, and the non-polar lipid molecules runs through the stationary phase along with the mobile phase. So what happens is here we can see the weaker interaction and stronger interactions. The first one is like the weaker, which depicts the non-polar lipid molecules as they can't bind with the silica gel which has the polarity, and the another one is the stronger interaction which has the highest polarity. Then this can be separated by releasing the knob which is present over here, and the eluted molecules are like this. The first eluted, like the green portion you can see here, is the non-polar lipids, and the another one is the polar lipids. Yeah, so this one is the first to separates the lipids on the basis of their uh, polarity. So yeah, the next one is the thin layer chromatography. It is as same as the adsorption chromatography, which works on the same principle to separate the lipids on basis of their polarity. So yeah, this is the TLC plate, which is composed of some materials. A thin layer of silica gel is applied to this plate, and the sample is spotted on this TLC plate, which is placed into a chamber which is filled with the solvent, naming the mobile phase. So what happens is the as the solvent rises with the capillary action throughout this TLC plate, the non-polar lipids runs through the same mobile phase, which moves farthest, and the polar lipids stays at the bottom, which has the most polar uh, capacity. So as we can say here, by the time the capillary action has increases and the molecules are separated, like the compound C maybe have the highest polarity and the compound A has the lowest polarity to it. So yeah, this can be used to separate the lipids. So now let's talk about the another one, which is HPLC, high performance liquid chromatography. So HPLC is the most commonly used method for the separation of lipids and their analysis. So and it gives the accurate analysis of the lipids. So yeah, so what happens in the HPLC is there is one HPLC column which is made up of sililic gel or silica gel in very compacted form. Another one is the solvent, which acts like the mobile phase, the pump, the detector, which gives the analysis of the data. So the solvents, like the mobile phase, is pumped with pressure through the HPLC column to the detector with a very high speed, and The sample lipids are injected to this pipe so that it can pass with the mobile phase. What happens is the HPLC column, the non-polar lipid moves faster out of the HPLC column, which is detected by the detector, and the another less polar molecules get stick to the silica gel, like separates them in that HPLC column, which is analyzed by the detector, which gives us a specific accurate data with their concentrations, like you can say the most preferable data with that. So yeah, this was the HPLC, and the HPLC must be used if you have the very ready equipments, as it may be expensive, but yeah, that's fine. So that's it from the lipids. Like we have covered almost some of the points, like structure of lipids, the linkages, the classification, the roles in the metabolism, cellular structures, etc. Along with that, we even studied about the techniques that had been used to study the lipids and their analysis. So yeah, thank you.